I am Lee Miller, President of the Society for the Neural Control of Movement. We're holding our annual meeting in Dublin this year, and I'm meeting here with Roger Lemon, our keynote speaker.
I am Roger Lemon. I'm Emeritus Professor in the Spinal Department at the Institute of Neurology at UCL in London.
Roger, you've been interested in the brain's control of reaching movements and use of the hand in particular for quite some time, and you've had a history of working with some of the luminaries in the field and become one yourself. I'm curious if you can tell us a bit about how you began that work and where it's taken you in the last number of decades.
I suppose I had a few sort of formative influences. Certainly one of them was working with Hans Kuipers in Rotterdam and then later in Cambridge, and he was, I think, probably one of the earliest of the sort of cadre of people who did refer to themselves as neuroscientists. This was sort of quite a new term. And why? How did he earn that? Because I think he was not just interested in the way the brain was wired up, the connections and so forth, but he was fundamentally also interested in how those connections worked in health and in disease. And so early on I got quite a very strong introduction to trying to make this relationship between structure and function, and particularly disordered function.
So it's worth noting, if I can interrupt, we've had a couple of sessions today and yesterday that in fact referred centrally to Kuipers' work.
Yeah.
It continues to be very important.
Yeah. And I suppose, and then going on from that, when I took the Sobel Chair in London in the mid-90s, of course, that is an institute that is heavily intertwined with the hospital next door, the National Hospital for Neurology and Neurosurgery. And of course, there you're so every day you're confronted with the kinds of disorders, neurological disorders that people that people suffer from. So, and those are the two things. I think actually trying to use the science to shed light on the disorders is a well-worn path, but it's not one that we should be changing just now, because I think one can see that the evidence is that it's only when we really understand the science of, for instance, of movement and control of movement, that we can really do anything meaningful to help patients with these different disorders.
What are the disorders in particular that are most relevant to the kinds of work you've been doing?
Because of my interest in the brain control of the hand, I've really focused on three types of disorders. One, a stroke, where undoubtedly loss of hand movement is devastating, almost as devastating, I think, as of course impairment of speech. Spinal injury, likewise. We heard today again this important data from Kim Anderson showing that spinal injured patients, of all the things that they would like restored, hand function is top of the list.
By a long shot.
And the final one, which is a more recent interest, but there's always been a long tradition in the Sobel department of interest in motor neuron disease. And I've become more and more interested in that because of the special ways in which the system that I've studied all my life, which is the direct cortical projection to the motor neurons of the hand, the evidence for this being implicated in the spread of ALS or motor neuron disease is now becoming stronger. So that's been of direct interest to both the science and the translational work.
Mm-hmm.
So so I think the way that I look at these problems is, and I think we see that at all levels. Right, you can go right down to the molecular, cellular systems, in whichever model you look. Any really significant piece of progress in terms of treatment, I think you can say that 90% of those treatments have arisen out of good scientific understanding of the problem. And sometimes, in the clinical world, we tend to see a pattern where sometimes the history, the background, where did this treatment come from, very rapidly recedes from people's consciousness once the treatment is established, of course, that all of the energy and funding and so forth goes into the treatment. That's absolutely right, but we shouldn't be doing that at the expense of forgetting where it came from.
Absolutely.
And how hard a lot of people worked at the problem in order to discover the treatment, which is now, having more and more benefit.
So you've given me a perfect entree into another direction I wanted to go with this conversation. The study of neuroscience generally and the control of movement has become extremely diverse with a huge range of available tools that we have these days in terms of computations, molecular genetics, and so forth that were simply not possible 50 years ago. Much of your work has, in fact, been dependent on working with monkeys and using the monkey model that has been in place for many years. Can you tell me more about its role in your work and how you view this work?
Yes, well, again, I suppose from the time that I spent with two leaders in the field, Bob Porter and Hans Kuipers, I was very impressed from the outset about the utility of the primate model. And why was that? Because I think the monkey has the engagement of a cognitive system, an animal that clearly understands the nature of a complex motor task, as well as the, if you like, the motor machinery to carry out those complex tasks. And it's the engagement of an advanced, highly advanced cognitive brain in a very skillful piece of skeletal-muscular machinery, if you like, that makes it such a good model for the human. And so, a lot of my work has involved the macaque monkey in particular. That is the best model that we have. But also, as we've gone along, we've understood that there are some very important differences between even the macaque monkey and humans. But we wouldn't have discovered those differences without doing the research in the first place. What I feel now is that most scientists who really want to advance their field are no longer content to be working with just one in just one area. And I myself have branched out a bit in doing, for instance, I've done quite a lot of work involving non-invasive methods such as transcranial magnetic stimulation. That's shown huge insights into how the human motor system works. But I would contend that without being able to have all the background information that we got from animal work, not just from monkey work, but from animal work generally, we would never be able to understand what TMS does to the human brain in terms of the way it can disrupt human brain function, the way it produces motor responses and so forth. All of that terrific insight into how the human motor system works has come about on the back of hard-won experimental knowledge from animals. But yeah, we are now using more techniques, and I think in the parlance of the current debate, many people who use animal models are being driven in the direction of saying you should be more interested in alternatives. And I say I push back against that and say these are not alternatives; they are complementary methods. Computational approaches, non-invasive approaches in humans, testing in vitro, if you like, testing across a whole variety of different animal models. To my view, these are all able to contribute pieces of information that the others can't. And so, of course, if you're really dedicated to getting an answer in a particular area that inspires you and interests you, of course you're going to use every tool that you can. But I think it's very short-sighted to say that we're going to arrive at an answer by saying, well, actually, one of these techniques we're no longer going to use. It's, and I'm afraid that the special place of the primate in this work, I think, is further and further highlighted by the fact that we are moving into a phase where, more and more the complex problems that we want to understand and the disorders that we most want to tackle are becoming more and more the ones that are most human. And by virtue of being most human, it's likely that the best model we have for the human, which is the macaque monkey, is going to be more and more used and of greater and greater utility. That's not to say that the molecular and genetic and other types of information that we get from other animal models is not going to be useful. That's going to be, extremely useful. I should give an example later today of how, molecular methods are used to identify pathogens in mice, and then we can see where those pathogens show up in the human brain and in the monkey brain, and you need all three to get to that point. But I think, again, what I would say is that most of the work is going to be done in the rodent, but there's still going to be a need for a carefully selected, carefully designed set of experiments in the non-human primate. They're not going to take place on anything like the scale that they were in the middle years of the last century, but there will still be some need in that direction.
And I believe, in fact, a central theme of your keynote talk later today is the comparison of different roles, different characteristics of the cortical spinal tract across species. It's worth noting one of the concerns, I think, these days is the applicability, the appropriateness of discoveries that are made in rodent models to human medicine. And I assume you would agree that understanding the differences we see across species with the potential then to extrapolate to humans makes this spectrum an even more powerful approach to understanding human and treating human disease.
Yeah, again, I think for all sorts of reasons, I try to avoid to get into a sort of us versus them attack on, say, rodent neuroscience versus primate neuroscience or human neuroscience, because I think in most areas you can point to very important advances made with all three, which you really wouldn't want to be without. But there is no question that when you want to, for instance, model how the integrative neuroscience system works, it seems to me, and that's when you bring in information that may well be related to basic cell characteristics, particular molecules, transmitters, these sorts of things, genetic influences, but when you need to see how the whole system works in a complex organism and use invasive techniques as well, then it's very difficult to come up with an argument that can find a better model than the non-human primate. So again, I see it as a complementary approach between these different models rather than let's do it here, let's do it there. We are experiencing a difficult period, I think, in terms of having to pull back a little bit from mouse models of just about everything and beginning to realize that some areas such as, say, depression and schizophrenia are likely to be products of a very advanced brain, such as the human, and where we may not get very far with animal models of those particular disorders.
It's interesting, we've talked about a large range of tools that are currently available. One of them that has become available in the last 5 or 10 years is the neural interface, or the brain-machine interface, that is increasingly being done with humans, having begun with actually both rodents and monkeys. It gives us a unique kind of insight into the functioning nervous system of either monkeys or humans, and is increasingly now being considered as a realistic therapeutic approach to humans as well. And I think it's an interesting, in some sense, full circle development in which these methods developed in rodent and monkey models are now actually being applied to humans, both to provide us with some basic understanding of the brain, as well as treatment of spinal cord injury and potentially stroke.
No, again, I would absolutely agree. I think that's one of the most recent and most exciting developments. I think if you talk to a lay audience about what is the contribution of the animal research that has been done, some of the figures showing restoration of movement to completely paralysed human patients, these are very compelling arguments, and even if you don't understand all the technology about how you get the signals and what you do to the signals before you put them back into the interface, you can't argue with the impact that having this available for a paralysed patient has a massive influence on their quality of life. So yeah, I agree. What could you say about the chances of us being able to develop brain-machine interfaces without doing the hard work in the animals first. I think that's the, you know, I point out when I'm talking to people about benefits of animal research, I do point out that, you know, there are several very famous groups around the world who are using brain-machine interfaces, including your own, and I make the point that in each of those groups, one of the leading lights will be someone who actually cut their teeth on understanding the technical, physiological, computational problems of recording this kind of data in an animal and then transfer that to the human condition. But you know, whichever group you go to, you'll see that one of the main drivers of the technology of applying this wonderful technique to human condition are people basically who have their background in animal neuroscience, and to me that speaks volumes, because it means that probably we wouldn't have been able to get to this without the hard work done in the animals.
If we look back a moment, in fact, to today's session, we had presentations from one group working with a patient who has a brain-machine interface to supply sensory information, and a subsequent presentation from a colleague of theirs who has done his work with monkeys and contributed to the human work. And I'm also struck with your comment about the compelling nature of this work with humans, whether it's the restoration of movement or the restoration of sensation. And in fact, this group at Pitt was visited by President Obama shortly after they had their breakthrough, in which they stimulated the brain to provide a sense of touch to the patient. And the climax of that meeting was when President Obama fist bumped with the robotic arm being controlled by the patient. It was quite a bit of media. It was very, very compelling.
Sure. And it also, I think, illustrates, beautifully there, something as, that's something as relatively simple as that, how much, complexity there is in attempting to restore such a simple movement. Again, that shows still, I'm afraid, the fact that we're still at a pretty early stage in understanding more complex movement. But for most of these patients, the provision of some basic return of some basic functions, including reach and grasp, can change their lives, and I think that's what drives us, keeps people wanting to do this research and wanting to continue to contribute. My own sense is that the demands on science will become greater because people will not, people now I think already do not accept that if they have a particular disorder, that they're stuck with that disorder forever. They expect treatments to come along, they expect better understanding of the disorder in the first place, and then eventually treatments that can either ameliorate it or even, hopefully even cure it. And there's a huge sort of expectation out there, I think, amongst the lay public as to what science can deliver. And so we're coming into a phase where it will be very interesting to see how well we perform against that expectation.
Yeah, I think the need for education is critical. It's also interesting, Elon Musk of SpaceX has now launched a new company called Neuralink, and in fact one of the members of that board is a member of our community and spoke here a couple of days ago. There's the possibility of a tremendous infusion of resources to the development of these kinds of interfaces for patients, and his goal is also to augment human performance in normal individuals, which strikes me as probably not a this decade kind of proposition, but if we can engage the fascination of the public with realistic expectations and a large amount of resources toward these means, it's hard to say where the limit may be.
Yeah. No, I think that's right. I suppose I should touch on what is the perhaps most sensitive area of this, which is that when the general public looks at the various advances that have been made, they of course make some kind of estimate of the cost of that work in terms of animal harms and suffering versus the scientific advance and hopefully also the translational benefits that appear in the clinic. And I think it's important to address that. Ethically, I think you make a decision at the beginning of your career that you either will or will not use animal models in your research. And to my way of thinking, that ought to apply to all animals, irrespective of speed. I don't see necessarily the monkey, the non-human primate, as being in a special class. I think the basic ethical argument applies to all animal life, and as distinct from humans. But where it is different to work with monkeys is that, of course, I think you have much more pressure on you to understand the nature of the harms that you are imposing on the animal in order to get this knowledge, and the responsibility you have for improving the quality of life of the animal in the laboratory. And over my lifetime, I have seen the most phenomenal change in that aspect of the work. So now I think if I look at in particular, the amount of training that goes into the people who care for animals, the way that the environment in which they live, their accommodation, the introduction of pair and group housing, which is of course an essential for such a social animal as a monkey, and then a whole panoply of technological improvements in the way that we do these experiments. So what has happened, I think, is really that we have managed to keep pace in terms of improving welfare, using what we call the 3 R's, to improve the welfare of the animals that we're using, has allowed us to continue to use these animals to derive useful scientific knowledge. And that's a twin aim that is not in any way mutually incompatible. That is, you can have good science that goes hand in hand with good welfare. And I think more and more we're beginning to realize that actually healthy animals that are psychologically well attuned to the needs of the experiment, generally deliver much better data.
Absolutely.
And better science. And so that's a sort of joint aim that we can aim at.
Yeah. Those of us working behavior with monkeys, that's been obvious from the beginning.
Yeah.
If the monkey's not happy and willing to cooperate with us, it will not work. Anything further you'd like to say, Roger?
No, I don't think so, other than to express my profound gratitude to the Neural Control of Movement people for inviting me to be the keynote speaker, and to come back to Dublin, which is one of my favourite places on the planet, and to experience what's been a really, I think, amazing programme, which sort of fills you with hope in terms of all the different things that are going on, and of course the demography, which is amazing. There's so many enthusiastic, young and brilliant guys at this meeting, so I'm absolutely convinced that neural control of movement is right up there in terms of long-term, future development and success. So congratulations to you as president for bringing us all here and putting on this great show.
Well, we're nearly at the end of what I think has been a very successful NCM 2017. I thank you to all the audience and your participation that's helped this be a great meeting. Many of you also contributed the sessions that we had this year, and I'll be looking to you to contribute sessions to next year as well. This is very much a community effort, and I appreciate all of your efforts in that respect. In 2009, we began our keynote tradition with a talk by Melville Jones, and since that time, we actually missed a couple of years right after that, but since then it's become something of a tradition, and really quite a nice way to close out our meeting, I believe. It's really a great pleasure for me to be able to introduce Roger this time, not the least because he hosted me and our family 10 years ago, something like that, for a summer in London, which we enjoyed greatly. It was, for me, very useful scientifically, and we also had a great chance to play football on the Hampstead Heath with Roger's lab. Our kids enjoyed competing with the old scientists and some of the young postdocs, including a few I see here. The NCM board members choose the keynote speaker after soliciting some suggestions for the speaker from the larger community. For next year, the nominations are due in November, so I encourage you to think about future possibilities. This year's choice was really quite easy. It was, in fact, a unanimous vote when the possibility of inviting Roger came up, and I contacted him, and he immediately said yes, so we were quite pleased to be able to have Roger as our keynote speaker this time. Both his bachelor's and PhD degree are from Sheffield University, and I believe both in physiology, although it wasn't clear on his CV. From 74 to 75, he was a research fellow at Monash University in Australia, where he worked with Bob Porter, one of the important people as part of his pedigree. And then he spent a number of years at Erasmus University, where he worked with Hans Kuipers. And also learned Dutch, I believe, which is more than I can say I did after 2 years postdoc in the Netherlands. My wife learned good Dutch, and maybe he and Roger can have a conversation later along those lines. In '94, he became the Sobel Chair of Neurophysiology at the University College London. And in 2002, he became the director of UCL Institute for Neurology. And in fact, they're working now to fill his shoes, which turned out to be big shoes. We'll follow that process as it continues. Roger is a fellow and past council member of the Academy of Medical Sciences in the UK. He was awarded the Fison Prize in 2015. His main research interest, as you all know, is in the control of skilled hand movements. In addition to its implications for the understanding of basic neuroscience, of course, it's also critical for understanding the deficits in hand movement that result from stroke, from spinal cord injury, from other degenerative diseases. Roger's experiments involve the use of monkeys, currently the best available model for human sensory motor system for controlling reach generally, but in particular, the dexterous use of the hand. Throughout this time, he's been a strong proponent of the use of monkeys, despite or perhaps because of the difficulties that this work is presented in the UK in particular, but also in the United States and Canada and Europe as well. This is an important part of what he's done at the Sobel. He's been actively engaged in the public dialogue on the responsible use of animals in biomedical research and in the importance of using monkeys as models of complex neurological and neurodegenerative disease. He's also carried out parallel studies in normal human volunteers and sought to apply the knowledge that he's gained from his work with monkeys to the effects of stroke on hand function in patients and to understand the process of recovery and to investigate therapies that might enhance that recovery. Roger will discuss the species differences in the organization of the cortical spinal system and their projections and how they are reflected in the different functions of the system across species and how the different models of motor control that people develop may profit from an understanding of these differences. The title of his talk tonight is Primate-Specific Features of Cortical Spinal Control. Roger.
Well, wow, what a lot of people. Can I just say a big thank you to the board and of course to Lee Miller for this invitation. I mean, it was very easy to give a very short latency response to the invitation that he sent, because of course I have attended NCM meetings over a number of years, and I know it's always a great meeting and always an exciting and interesting time, so it really is a great honour to be asked to give the keynote lecture. Now, the next slide I'm going to show you is the slide I usually put at the end of my talks, and it looks something like this. Because as you just heard from Lee Miller, there is a tradition of considering the sensitive nature of the primate in a rather different context from the use of other animal models. And I myself am persuaded of the fact that if we look at some of these are the clinical benefits, but of course we shouldn't ignore the major benefit that has come from the use of monkeys in research as being our ever-expanding knowledge about the brain, and including the human brain. That comes first, and some of that knowledge has then been developed into the use of treatments and medical benefits, and some of those are listed here. And I thought I would just illustrate this point by talking to you about the brain prize for 2017. And this has been awarded to three very clever people. All of them happen to be working in the UK: Peter Diane, Ray Dolan, and Wolfram Schultz. And this prize has been particularly awarded for an understanding of complex neuropsychiatric conditions. So it's quite interesting to see how this triad of clever guys work together. So Peter Diane is a computational neuroscientist, Ray Dolan is a brain imager and a neuropsychiatrist, but actually they're all dependent. The whole story began with some very important basic work that I'm sure you're all familiar with on the reward system and the involvement of dopamine in that system that was carried out by Wolfram Schultz. I think it's a very good example of how trying to understand these complex disorders starts with very good basic science. And I do not defend the use of the monkey model per se. I defend the use of this model to do high-quality science and thereby produce more knowledge and more translational benefit. So, got that little thing out of the way. Now I can talk about the lecture. The lecture tonight is going to be about the way the corticospinal system is adapted in different species. And in particular, I want to try and make the point that this pathway that runs from cortex to spinal cord has been exploited by many different species in many different ways in order to make them all what they very, very much are, which is very successful occupiers of their particular ecological niche. So I will talk about that at the beginning. I will come to focus on one particular component of that system, that's the corticomotor neuronal system, and then I will move on to three analysis of three other features which are particularly interesting in primates, including the distribution of the fibers within this system, the spike duration within the neurons that make it up, and most recently the expression of a particular protein, the fast potassium channel protein KV3.1b. And then I'll sort of feed this into if you like, new light on an old idea, which is the concept of evolution and dissolution in an advanced nervous system, which is becoming more important as we see that many different disorders impact ultimately on skilled use of the hand. As ever, I should thank a large list of members of my lab, or what was my lab, and collaborators at UCL and in various other important places. They're all listed there, and I thank them for their contributions. So I think we've heard several times during this meeting the concept of a conserved system, and we can accept that many mammalian species have a model which is built on and is added to in different directions. So one good example of that, of course, is the concept that the map of motor neurons within the spinal cord, the cervical spinal cord, the cervical enlargement, we find groups of motor neurons to deal with innervating flexor muscles and extensor muscles in different places, and we find flexors and extensors concerned with trunk, shoulder, forearm, and hand located respectively from medial to lateral. This basic pattern is one example of a highly conserved element of a motor system. And another example that you'll be familiar with is this diagram from a paper by Sten Grillman and colleagues where they looked at the way that different behaviours like locomotion, posture, and saccadic eye movements are driven through a system involving the cortex striate and pallidum. So these are examples then of the conserved system. I should be talking about the corticospinal system, which also has some conserved functions, but in particular it has also some rather interesting variations according to the particular species that you look at. So, in looking at across the mammalian kingdom and the way in which the corticospinal system functions, you discover, as I say, that different species have evolved to use this system in rather different ways. Just as you might use a communication system inside a computer to link up different parts of the system and to actually carry out different processes. So, for instance, one of the processes that the corticospinal system is very much involved in is actually nothing to do with movement per se, although it is related to it in terms of reafferents, but its main job is to control sensory afferent input, and of course most of that is occurring through the mechanism of presynaptic inhibition. We had a very interesting workshop on that earlier in the week, and that involves not only sensory input from large fibre systems, but also nociceptive inputs related to pain. That's actually a property of the corticospinal system that is shared by nearly all mammals, but is, I think, received far less attention than the so-called motor properties of this system. Another function is the selection and control gating of spinal reflexes and the supraspinal modulation of central pattern generators within the spinal cord. True motor functions do exist, of course, through direct and indirect activation of motor neurons and the inhibition of motor neurons involving a variety of inhibitory interneurons. But there are other interesting features in this system as well. A few years ago, Alexander Kraskov and I first showed that some corticospinal neurons can behave as mirror neurons, so they not only are involved in the generation of their own movement, but they monitor the movement of others. Peter Strick and his colleagues have spent quite a lot of time looking at corticospinal innervation of non-motor structures like the kidney, and discovered that there are a very large number of neurons within the corticospinal system that seem to be related to these visceromotor activities. And finally, another important area is during development, the fact that the normal development of the corticospinal tract seems to be important for allowing, for instance, motor neurons to develop their complete adult structure. Without that long-term trophic influence, the spinal cord doesn't develop normally. So quite a lot of different functions are employed to different extents and different degrees in different species. So I'd just like to take you through a few examples of those variations, and I'm going to begin with an elephant. Seems believe it or not, people have actually looked at the pattern of termination in the elephant. These are elephants that unfortunately suffer strokes in the zoo, and then a very enthusiastic anatomist goes off and begins to dissect out the spinal cord and brainstem in an elephant, which is not at all easy. But what's really interesting is the results of these structures show that most of the fibres that descend from the cortex actually don't even reach the spinal cord. The great majority terminate in the facial motor nucleus, and of course this is the nucleus that innervates the trunk. In the rat, what we have is a very good example of this system that seems to be particularly devoted to control of afferent input. We have a heavy corticospinal termination in the dorsal horn. You can see that here, with less dense projections into the ventral horn. And I think there's a literature on corticospinal lesions in the rat, which suggests that most of the problems are sensory motor rather than purely motor problems. But it's interesting that in comparison to the lack of direct projections to fall in motor neurons in the rat, there are direct cortical projections to another group of motor neurons which are very important to the rat's vibrisal system. So the facial motor neurons that are involved in the whisking function of the rat do receive direct corticofugal projections in this species. We know a little bit less about this system in the mouse, although there's a lot more very interesting information coming out. And but what we know is that it's a system that is largely directed towards the dorsal horn, not so much to the ventral horn. We do know actually from the work of the late Laura Alstermark that stimulation of the corticospinal system in the mouse gives rise to relatively long latency oligosynaptic effects. Looking at some primates now, the spider monkey is a particularly interesting one because it has this massive and very powerful tail, which it uses to hang from as it moves around. It's an arboreal monkey, and interestingly, this tail has a glabrous, non-hairy skin, so just like your fingerprint skin, which it uses obviously to explore the branches and other articles that it uses to suspend itself on and leave the other four limbs free. And it's interesting that in this species there are direct projections from the cortex to the tail motor neurons present. Peter Strick, in a study where he looked at the projection to the forelimb motor neurons in this monkey, the capuchin or cebus monkey, showed that there was a very strong projection down into the most lateral parts of the ventral horn. This is where the muscles, the motor neurons innervating the intrinsic hand muscles are located. And that is an interesting discovery because this particular monkey, the capuchin monkey, is a very skillful tool user, both in the wild, but particularly actually in the laboratory. It's been shown to do all sorts of be able to do all sorts of clever things. It is definitely a good tool user, and it happens to have this very strong projection. We'll see that other New World monkeys lack this projection and are generally not recognized as being natural tool users. The first ever report of direct projections from the cortex to the motor neurons of the spinal cord is actually hidden away in a report by Layton and Sherrington in 1917. And they saw degenerating terminals after lesions in the primary motor cortex of the chimpanzee. They found a very extensive number of evidence for degeneration down there in the ventral horn. So this is the animal that has the animal that we know most about in terms of the manufacture and use of tools, which is again it has very considerable variety of skills in that area. It also has this very strong corticomotor neuronal component. Another area, of course, of great interest in terms of cortical projections is related to vocalization, and it turns out that there's good anatomical evidence in the human system for a projection from the orofacial motor cortex to the nucleus ambiguus, which is where we find the motor neurons of the intrinsic laryngeal muscles. And that, of course, is not surprising. So now some clinical people are now stimulating directly this area and are able to begin to see whether they can find direct electrophysiological evidence that these are also corticomotor neuronal projections to the motor neurons of the intrinsic laryngeal muscles. There's no direct evidence for that yet. By contrast, in the macaque, it's interesting that there are no direct projections to the nucleus ambiguus, but there are, of course, very strong projections, as we heard from Stuart Baker earlier, from these areas into the adjacent reticular formation. Now, in trying to look at these variations and seeing what sort of patterns might emerge, of course, was first undertaken by on Skype, was looking at the question of whether species differences could be related to these corticospinal patterns of variation. And he did a very extensive study looking at the differences in different species. This is a rather famous picture of his, where he showed that if you looked at a possum, cat, rhesus monkey, and chimpanzee, the main thing that you see is a ventralward shift in the pattern of termination across these different species.
And in particular, it's striking that this projection to the ventral horn is very much more marked in a great ape, which is the chimpanzee, compared to that in the rhesus monkey. And now, of course, I think from the physiology that have now been carried out in quite a lot of different species, it turns out that we see that, in fact, these direct CM projections are only found in the more dexterous primates, not in just every primate. So, for instance, the marmoset doesn't have any direct projections of this kind. The Cepus monkey does, and of course the rhesus monkey does. So that brings me on to this corticomotor system. It's, as I say, a component of the corticospinal tract. It involves these direct projections. Mostly, we think now, again, this is work from Jean-Albert Ratelot and Peter Strick in Pittsburgh, showing that most of these projections seem to come from primary motor cortex and from area 3A. The primary motor cortex ones terminate on a whole bunch of different motor neurons, a whole bunch of different muscles, and probably in humans these are extremely widespread, the number of muscles that receive this direct cortical projection. But nevertheless, it's a graded projection, and the projection is strongest to what you might call the most voluntary components of your musculature, so the face, lips, hands, and feet. And so what we're looking at is particularly interested in these terminations here. This is an anterograde peripheral nerve tests that these problems are not a result of the lower motor neuron problem. These are definitely something to do with the, if you like, upper motor neuron input to these particular muscles. And the distinctive feature of patients at this time is that if you ask them to write, they will tend to write holding a pen or a pencil between their ring finger and little finger, because they still have some strength and control there, whereas that has already gone between the thumb and the index finger. So, of course, this is an interesting pattern of effects, because we know that the CM system is particularly focused on the hand compared to, for instance, wrist or elbow muscles. But we also know from detailed investigations that were actually carried out by Charles Phillips over 50 years ago that there's a greater CM input to thenar muscles than hypothenar muscles. That's been confirmed by studies that we have done using spinal triggered averaging. But basically what you have is that the largest CM input is directed towards the thenar muscles, the intrinsic thumb muscles, and although there are also large effects in the hypothenar, they are generally weaker than the thenar ones. So it's just possible that difference is enough during this fast-moving stages of ALS to result in a weakness in the thenar compared to the hypothenar, and to create this interesting phenomenon of the split hand problem. Now, at the same time as Andrew Eissen's been making these observations, a group of neuropathologists in Ulm in Germany, led by Heike Brack, have developed an interesting theory about how ALS spreads through the nervous system. They're particularly focused on these are post-mortem changes that they see in many different, 22 different brain regions in 76 patients who are confirmed to have died from ALS. And in their hypothesis, the first stage of this disorder is a prion-like propagation of a compound called TDP43 that seems to spread from these neurons through their axons. And it spreads through the axons because the protein is solubilized in the axon, whereas in the cell body it can be fixed. So the fact that they are concentrating particularly on layer 5 neurons with long projections could explain the fact that, of course, in many types of ALS, not necessarily all types of ALS, there is evidence for both the so-called upper motor neuron, i.e. the cortical neurons, and the lower motor neuron, i.e. the spinal neuron. In these neurons, the pathology shows that this TDP43 material is converted into a sort of scheme-like aggregate within the cell body, and that's good for the cell. It seems to somehow isolate it and prevent it hurting the cell any further. But unfortunately, it can also be transported through the axon, and it can be shown that, in fact, this pathological compound then affects the lower motor neuron. And unfortunately, the lower motor neurons don't seem to be so resistant to the existence of this TDP43 within their system, and they suffer and they die. So the solubility in the long axons is particularly interesting in view of the fact that maybe this disorder begins at a cortical level and then spreads through the rest of the motor system according to the pattern of innervation of the most direct system, the cortical motor neuronal system. So under that idea, you'd expect the muscles that are under most cortical control to be the ones that suffer first, and that's true. So vocalization is affected, hand control is affected, and even in locomotion, ALS patients typically report their first major problem is doing very, very skilled forms of locomotion, such as skiing and climbing. So, what's the evidence then? Another bit of evidence is that it turns out that the motor neurons that receive no CM input, and those are the neurons of the onus nucleus that control the sphincters, and the oculomotor nuclei, these receive no cortical direct projection. Interestingly, these motor neurons are the ones that survive longest during the destructive course of motor neuron disease. It also turns out that cells with very short axons, such as spinal and cortical interneurons, are also relatively resistant to this TDP43 pathology. And it also turns out that this pathology, of course, affects not only the cells, but also the glia, and particularly the oligodendrocytes that support these long fibers as they travel through the brainstem and spinal cord, also show these inclusion bodies. So, mechanisms that can understand how this pathogen spreads through the system and how it's converted to these inclusion bodies rather than leaving it soluble is quite exciting in terms of presenting a new therapeutic target for ALS. So let me wrap up. Although the motor system in different species shows conserved characteristics, I hope I've persuaded you that the corticospinal system exhibits species-specific features as well. In dexterous primates, the most conspicuous difference is the presence of these direct CM projections. However, there are other interesting and possibly related differences, including production velocity, spike duration, and the expression of KD3.1b. Primate-specific features like this might explain the particular vulnerability to disorders such as ALS, with early involvement of layer 5 neurons and the loss of CM connections. And finally, species-specific features of corticospinal organization, including major differences between the mouse and the primate, need to be taken into account in understanding this pathology. And there I'd like to finish and thank you for attending this lecture. As we say in English, after the Lord Mayor's parade comes the corporation dust cart.